This video is a continuation of our discussion of pharmacogenomics, or some people call it pharmacogenetics. So as a reminder, pharmacogenomics is just the idea that genetic variations in people that take drugs is going to influence the way that those drugs impact them. And it can happen in a few different ways. So I wanted to back up a little bit and talk about genetic variations, how they exist, how they're measured, and how we're going to put them to use. So the first idea is you've got to figure out what those genetic variations are. And the majority of differences between people, so if you just go into a general population and you look at variations that you're finding between people in that population, the majority of them are just single nucleotides. So it's not huge, massive changes to their chromosomes. It's single base pairs. So between individuals, you might be going along in your DNA samples that you've taken from, at this point, thousands of humans have had their genomes sequenced to some degree. And so we might be going along and saying, okay, they all have a T at this position, they all have a G at this position, an A at the next one. And then we get to the spot where there's some variation. Some people have a G, some people have a C. You don't necessarily call this a mutation because you can't exactly say the G or the C is the good one or the bad one. It's not always clear like that. All we know is there are differences. And so the term polymorphism, it just means many varieties. And so you have thousands and thousands of these SNPs sprinkled throughout the genome. And the hard part of all this, when the human genome was first being analyzed, was figuring out, all right, we know these variations are there. What kind of effect are they going to have? And they can have a few effects. We're focusing on the pharmacogenomic side of this, right? How do those SNP variations in the genome affect the way that people respond to drugs? You can also ask questions like, what diseases does this predispose a person to? So, for example, this genome-wide association study, or GWAS, that takes those SNPs and asks, are people more or less likely to have a certain condition to begin with? And so this all comes full circle because your genetics can determine what conditions you're more likely to have, and they can determine how you're going to respond to treatments to those conditions once you already have them. Right? And the goal in all of this is to inform doctors and pharmacists and anyone else working in these fields with the information about which SNPs are going to have the most effect and which of their patients have those SNPs. So in order to make all of this work and put the information to use, we're going to have to know about these genetic variations in the patients that are getting a treatment. And the easy way to do this is say, all right, once we have a diagnosis, then we search those relevant SNPs, sequence them, and try to get data back to inform what drugs we're going to give and maybe what dose we give them at. So circling back to some more fundamental biology, how can a single nucleotide variation have such a big impact when there are about 3 billion nucleotides in a genome? So just as a reminder about some basic genetics, DNA sequence determines RNA sequence, and RNA sequences that you're seeing here determine amino acid sequence. And so these codons from RNA determine the amino acids that make up proteins, right? Phenylalanine, leucine, isoleucine, these are the amino acid choices that cells have when they're building proteins. And just as a reminder, those different amino acids have very, very different properties. And so it might seem like a minor thing, but if you change one nucleotide in a gene, you might change a small hydrophobic amino acid like alanine to a very large charged polar water-soluble amino acid like a lysine. That single amino acid change, if it's dramatic enough, can disrupt the function of a protein. And that disruption might be relatively minor, like slightly changing the speed at which an enzyme carries out a reaction, or it could be a huge change, like the protein is unstable now and isn't even going to be produced. And the changes can go in either direction. More active, less active, more protein produced, less protein produced. And you really don't know exactly what effect the mutation is going to have or the SNP is going to have until you test it. And so this entire field requires developing a very large database with a lot of information. So let's think through some possibilities of how this could connect to pharmacogenomics. Remember, enzymes and receptors and all sorts of other protein structures, these are most commonly our targets for drugs. Okay, so if a single amino acid changes the structure of a protein, it could change how that protein interacts with substrate molecules if our protein in question is an enzyme. Right? Drugs are almost always the substrates for enzymes at some point because that's how they're metabolized and eliminated from the body. If you didn't have enzymes degrading the drug, then they would just never go away and you wouldn't even have a half-life. It can change the way that proteins interact with ligands. Remember, a ligand is just any chemical signal, like a neurotransmitter or a hormone or a paracrine signal. And that would be the case if our protein of interest that contains the SNP is a receptor of some sort on the surface of a cell. Okay? And of course, for the pharmaco part of pharmacogenomics, it can change the way that the drug itself interacts with the protein. All right, so we have a lot of different possibilities here. And this setup is showing you, remember, our levels of protein structure. We start off by building a primary structure out of a sequence of amino acids. We fold those amino acids into secondary structures like alpha helices and beta sheets, and we further fold those structures back on themselves to make complicated three-dimensional structures that we call tertiary structures. And if multiple proteins interact with each other, we can call those quaternary structures. And the point is, a single amino acid change back here in the primary structure can cascade forward to slightly disrupt secondary and tertiary and quaternary structures, and that disrupts potentially the function of the protein. It's also possible that the mutations enhance the function of the protein. Again, it's a case-by-case basis. All right, so let's use enzymatic reactions in the liver as an example. So when someone takes a drug, your liver has to be prepared to metabolize any drug that you can throw at it. And because of that, there are an enormous number of enzymes that your liver keeps produced at all times, ready and waiting in case some sort of toxic molecule or drug enters the system. And in general, these are split into two phases, phase 1 and phase 2 reactions. And the details of this aren't really critical for us right now. That would be a biochemistry topic. But the idea is phase 1 reactions make the drug reactive in some way. Right? So we add functional groups like alcohols or sulfur groups to make the drug reactive so that it's easier to conjugate them with either acetyl groups or glucoses or anything that is highly polar and water soluble. Because the end goal of all this is we want the molecule that leaves the liver to become as water soluble as possible. The reason for that is all these different excretion systems, they find it easier to secrete water-soluble drugs because they dissolve in the blood or plasma more easily. By far the most important organ in that process is the kidney. And so if the liver can make your drug into a modified version of that molecule that's got a higher water solubility, it's easier to excrete, which means that you would expect its half-life to decrease, right, and be removed faster. Now, for medical reasons, decreasing the half-life isn't necessarily a good thing. If we're giving a drug that we think is going to help a patient, it would be a good thing to keep it in the blood for a longer period of time. And so the critical part of this is we want to know if the half-life is accurate or not. It's not necessarily about being longer, being shorter. We just want it to be consistent so that when a doctor or a pharmacist prescribes a certain dose of a drug, that we know what to expect. All right, so as a reminder, half-life means if we're giving the drug right here and levels increase very quickly, half-life is just the amount of time that it takes for the concentration of the drug in a person's system to cut in half. And so in this example, they show us a data point where the blood level is 50. Half of that is 25. And so the time that it took on our x-axis down here to get from 50 to 25, that would be our half-life, which looks like approximately 8 to 10 hours in this case. All right. If you then dose again and levels rise, you keep having this oscillating function. But at some point, after about 4 half-lives, you hit stability where the oscillations stay consistent and you'll hover between a high and a low range. If someone had a mutation in the enzyme that's metabolizing this drug and the enzyme slowed down, we would expect that this stable point would now become higher, right? Because it would take more time to metabolize the drug and the steady state level would go up. If the enzyme became faster, we would expect that steady point to come down because it's being eliminated more quickly. And so in this example, we're not really talking about whether the drug works or not. We're talking about how fast the drug's eliminated and how that determines the appropriate dosing scheme. All right, so as some examples of the number of different enzymes that the liver uses in these cases, the most common family by far are called the CYP enzymes, and they're often called cytochromes. So all of these different CYP designations in this column are different CYP genes, and the next column are alleles, which are variations. So these are basically known SNPs or variations in those CYP genes, and this is their percentage in a European population that was measured, and the enzyme activity and the test drug is asking the question, okay, does that variation either increase or decrease metabolism? And if so, for what drug? So you can see just in this table alone, which is not by any means an exhaustive list, we have a lot of different drugs, everything from nicotine and caffeine to really important clinical drugs like warfarin. All right. So if these numbers are substantial, right, anywhere from a few percent up to 24, 15, 30 something percent, these are not trivial amounts. That means that we're going to use the example of warfarin as a way to illustrate this point. If these variants are detectable in 19% of people, it would probably be important that physicians and pharmacists know whether someone fits that genotype or not, because 1 in 5 people have it if they're going to show decreased enzymatic activity when it comes time for the liver to remove that warfarin from their blood. Okay, so to try to tie all this together, a few things. First of all, I'm using the liver as an example for this, but this map shows cytochrome expression across the body. So the liver is by far the most important organ for metabolizing drugs, but these CYP enzymes are also found in many other locations, everything from stomachs to spleens to kidneys to hearts. And their expression tends to be lower there than the liver, but we can't treat this 100% as a liver thing. All right. The other thing to remind you of is these mutations or SNPs can have a wide variety of different effects. The table that we just looked at shows it as simply an increase or a decrease, but it's slightly more complicated than that. So you could have everything from a SNP that eliminated production of the mRNA altogether, in which case you wouldn't even have an enzyme and there would be zero metabolism. That would be an extreme case, which is not very common in the population, but it happens, right? On the middle ground, you can have an enzyme that is unstable. So you're not necessarily slowing down each individual enzyme. You're just going to have less of them because their half-life goes down and the amount of those enzyme copies in the liver would decrease. So that would lead to decreased metabolism, right? On the high end of things, you could have higher levels of the enzyme. So that is effectively the exact opposite of the unstable category. There you have additional copies, which should lead to increased metabolism. All right, and the last category, altered substrate specificity. This is different altogether. So the other ones that I've highlighted here, they're not changing what is being metabolized, they're just changing how fast. If you mutate the binding site for the substrate, you could affect what it even recognizes. And so in this case, it could eliminate some drugs as being recognizable by the enzyme, and it could add some other drugs by modifying the shape of the active site where those drugs stick to the enzyme, right? And then these examples down here, CYP with these designators after them, those are just specific SNPs that they're using as examples of those particular categories. All right, so as an example of this, I'm going to use Warfarin, which is a drug that's used as an anticoagulant in clinical settings. So when someone takes Warfarin, there are a handful of things that are going to happen. Since Warfarin's an anticoagulant, it has to have some sort of target protein that's in the coagulation cascade that causes blood clotting. And its target is this VKOR enzyme. And VKOR is necessary for activating vitamin K. For reasons that are probably outside of this discussion, vitamin K is necessary to turn on clotting factors in the blood. And so if a person slightly decreases their level of vitamin K, it will make them less likely to clot. That's a good thing for someone who has a clotting disorder and we want to decrease that activity, or someone who's going into a major surgery and they're going to have a surgeon cutting through their blood vessels. That has a tendency to stimulate clotting, and we just want to make sure that it doesn't go out of control. All right, so this would be the actual effect, the desired effect of the drug. And then up top, we're looking at metabolism. And what I thought was interesting about this is your liver can actually metabolize warfarin using 3 different enzymes. So all of these different CYP enzymes are different genes, slightly different versions of that enzyme, and they convert warfarin into slightly different byproducts. Notice that they all have an OH, a hydroxyl group. The reason for that is that's increasing its reactivity and water solubility, but it's putting the OH at different positions, right? We can add that oxygen and hydrogen to many different locations here, and that's the variation among those enzymes. So the level of complication is sort of growing at this point. We have a drug that can be metabolized by multiple enzymes, each of which has many possible SNPs that you could find throughout the population. And so trying to figure out which of these enzymes and which variations are going to have an effect can get pretty complicated and is going to require a fairly large data set. Okay, so this was an interesting paper because they're actually looking at 2 separate things. They're looking at the effect of variations in a CYP gene that affects metabolism and breakdown in the liver. And they're also looking at the VKOR gene that codes for the protein that's warfarin's target. And the question they're asking is, do genetic variations in either of those genes or both of them change anything about patient response to the drug that's significant enough that we should be paying attention, right? This is a systematic review and meta-analysis, which means they took papers from many other publications from other labs, and we're trying to collect them all together, do a statistical analysis, and ask, do they concur in giving us the conclusion that these 2 CYP and VKOR genes matter and that we should be paying attention to them when we're determining dosing schemes for warfarin. All right. So the conclusions. These tables, these forest plots, when you read these, right, each one of these rows on the side is a clinical trial that was conducted independently. And since this is a meta-analysis, they're taking all of the clinical trials that they can find in the published literature and summarizing them by doing a statistical analysis that asks a pretty simple question. Does the genotype, in this case CYP2C92, is the CYP variation, and there's also a 3. Those are 2 different SNPs that are associated with that gene. Do those genetic variations change something called stable dose? Stable dose is what we were talking about earlier with half-life, right? How much of the drug do you need to give a patient before they reach a stable therapeutic level that you want them to be at? And so the way we read this is, if the data come out shifted to the left of 0, that means that a lower dose is needed to get them to a therapeutic range. And if it came out to the right of 0, it would mean that you need a higher dose. Right? So in this case, we got an across-the-board decrease in the amount of stable dose that was necessary to get these patients to their therapeutic range. And that makes perfect sense, because if you go back and look at the data for CYP2C9, people with this mutation have slower metabolism of warfarin. And so if their metabolism is slower, that means the drug is staying in their system for longer than a normal person. And so we expect that we would need to give them a lower dose. The significance of that is their doctor or pharmacist needs to be aware of that, because if they ignore that genetic data and give them a normal dose anyway, it means that they're going to have a higher blood level than a person without that genetic variation would. On the other side of this, same thing, but for VKORC1. In this case, these people need a higher dose to get stability. And the reason for that is this mutation makes the drug slightly less effective, right? And so you have to give a higher dose, not because it's being eliminated from the body in a different way, but because it's changing the dynamics of how it binds to its target protein. Right? And so this was, I thought, a really illustrative example where you can see two things at the same time. One set of genetic variations, right, the CYP enzymes, change metabolism and half-life and change the amount of drug that you expect to find in the patient's blood. And the second enzyme and its genetic variation change the way the drug interacts with its target, but you still get a change in stable dose. So two completely different genes and two completely different mechanisms are sort of feeding into each other to give you this overall effect. Right? So to sort of wrap it up in summary, that brings us back up to why this matters. Right? Why do we collect genetic variation data? Why do we need to organize it into a database? Right? This DB-SNP database that's managed by NCBI. Their goal is to gather as many of these genetic variant SNPs as possible, connect them to information like we were seeing in this paper about pharmacogenomics and about risk for different diseases. And the hope is that when a patient is diagnosed, as collecting this genetic data becomes cheaper, that doctors will be able to get SNPs sequenced for their patients and use that as a method to determine what drugs they should get and in what amount. All right. Hope you found this interesting, and look forward to discussing.